So thanks, Robbie, for organizing this workshop. Yeah, always nice to. I was checking. We've published 13 papers together.
Only 13. My impression was that at least 100.
Yeah, it sometimes feels like it in a good way.
In a good way. Yeah.
So I mean, I thought I would just discuss. Well, one of the last projects that you did. Well, you started while you were in Liverpool, but we only actually published it last year, which is the Cellage database and systems biology analysis of cellular senescence. So I mean, I guess you know already of our human aging genomic resources, which is something I started as a PhD student as a side project. For those who don't know, it was not part of my PhD thesis. It was just something I became interested in. I was interested in longevity of species and also genes associated with aging. So I kind of start keeping track of them on an Excel spreadsheet initially, and then I just decided to make databases out of it. And this really has evolved into this collection of resources. And so in particular, I think the GeneAge database reflects how much we've advanced in the field in terms of genetic understanding of aging. I'm glad many of you have used it. For those interested in biology of aging, I also have Twitter and Facebook pages where whenever I see something interesting, I tend to post it there. And so I think in terms of the GeneAge database, called the Gerontome, and oh, by the way, I really like the title of the workshop, Gerontomics. I think it's really cool. So I think it really reflects how much we've advanced in the field. And I mean, the last version, this last paper with Robbie as well, with Vadim, who's online as well, and others. I think it really reflects this huge advances in terms of understanding of genetic basis of aging. And really the challenge now is to keep track of everything. So it's good that we still work together on this. So in terms of, I mean, one of the databases or one of the topics is cell senescence that we've also been focusing on. I mean, cell senescence, I mean, I tend to define cell senescence as cells that should be dividing, but just irreversibly stopped dividing. And, this was identified by Lena Hayflick initially 60 years ago now, in vitro. But we also know that senescent cells also accumulate in tissue. So me and Jean Passos, he was in Newcastle at the time, now he's in the Mayo Clinic, we did this review. And I should say, he made these really nice figures. I'm not very good at making figures. But the hypothesis is that, with stress and with time and with age, you have this accumulation of these non-dividing senescent cells. And these non-dividing senescent cells, they also secrete inflammatory cytokines, called the SAFs, senescence-associated secretory phenotype, which contributes to inflammation and tissue dysfunction. There's a lot of interest in it at the moment. I mean, historically, I actually did my PhD in cell senescence and telomeres, and then I didn't work on it for some years. But then after particularly the Van Dersen papers showing that ablation of senescent cells in mice extends lifespan and ameliorates, well, not everything, but some facets of aging, I became interested in it again. And I mean, I'll come back in the last slide actually to my own ideas on senescence. But I think there is evidence now that at least in rodents, they play some role in aging. And so there's a lot of companies also developing drugs, in particular senolytics, to target senescent cells. Unity Biotech, for example, funding from Jeff Bezos, the wealthiest man in the world, being the prime example. But there's quite a few companies now working on senolytics. So it's quite a timely topic. I would say it's a fashionable topic, cell senescence. And so we did this database, which, as I said at the beginning, was started by Robbie. I mean, it was something started several years ago while we were still in Liverpool. And the idea was to catalog genes associated with cell senescence in the same way we catalog genes in GeneAge. So by looking at genetic manipulations, and if you do a genetic manipulation of a gene and it induces or prevents senescence, then we could also classify genes as senescent. Well, pro or anti, or actually I think it's promoters or inhibitors of senescence. So the current version has 279 human genes, but I should say the next version will have, I think, more than twice actually, those numbers. And as I said, you can have different types of senescence and also different promoters or inhibitors of senescence. This is a photo from my PhD, so from those are young fibroblasts, and these are senescent fibroblasts. Senescent fibroblasts, as you can see, they have quite a diverse morphology. So you can have elongated cells, big cells, non-senescent young fibroblasts tend to be more homogeneous. So as I said, you have different types of senescence. You can classify them as promoters or inhibitors of senescence as well. I think I have a plot on that actually. Yes. So this shows you like the distribution of genes. So we have inhibitors of senescence, 121, inducers of senescence, 153, and there's 5 cases that are unclear or difficult to define. And then you have the different types of senescence, which is replicative senescence. But then you can also induce senescence by stress or by oncogene. So there's different types of classification of senescence, although again, sometimes a bit of a gray area, I should say. It's not. But there's different types of senescence. And this is reflected in the database. And then we did several analyses based on this genes associated with senescence. So the usual take of the databases we have is when we have a new database, we do like a first pass analysis of the data, including evolutionary analysis, network analysis. So I'll briefly mention this. So, we did a functional enrichment analysis. So this is just top categories, signal transduction, P53 mediated, not surprising, given that P53 is a major mediator of cell senescence, aging as well, and other aspects like cell growth and so on. So, categories by and large you would expect to find. Then we did an evolutionary analysis. We show here that, so this is the percentage of human orthologs in the genome and those in cell age, which you see in red. And there's a greater number of orthologs in cell age than expected by chance. That's basically what this is showing in primates, in mammals, so mice, rats, and monkeys, which I kind of expected because cell senescence is a relatively well-conserved process in mammals, as far as we know. But you don't see the same in, for example, worms or flies, which makes sense because worms or flies, as far as I know, they don't have senescence. Well, they certainly don't have replicative senescence. It's normal that you don't see these same pathways in worms or flies. So cell senescence is quite specific, of course, to organisms that cells proliferate, and worms and flies, they're mostly post-mitotic as adults. So their cells don't divide as adults. So they don't really need cell senescence. So we also looked into the senescence cells senescence genes in different human tissues. Now, I thought this was interesting. So this is using GTX data. So what you have here are various human tissues, adipose tissue, gland, blood, etc. And then you have inhibitors of senescence, and whether they're going up or down. And then you have promoters of senescence, whether they're going up or down. And I mean, in inhibitors of senescence, there's not much of a pattern, but in terms of the promoters of cell senescence, there is an overrepresentation of genes of these promoters of senescence being overexpressed with age in human tissues. Now, as you can see, it's not all tissues, it's not even most tissues, but it's more tissues than expected by chance. So we could say that, you do see these genes promoting cell senescence more overexpressed with age or in more tissues than you'd expect by chance. With some exceptions, I mean, the uterus is quite a striking exception. I mean, there's some other data on this as well. But by and large, you do see these patterns of cell senescence promoters being overexpressed with age in human tissues. I mean, we also derive gene expression signatures. That's something we've done, well, for other processes like aging, like we did for caloric restriction. I think Robbie was involved in that, the genome biology paper 2015 as well, where we did gene expression profiling of caloric restriction. So we did a gene expression profiling of cell senescence. So basically, this was a meta-analysis. So we took 20 data sets from GEO of cell senescence, so gene expression data, and then you combine them in a statistical meta-analysis framework, and then you derive a signature, gene expression signature of cell senescence, which is composed of over 500 genes overexpressed in cell senescence and over 700 genes that are consistently underexpressed in cell senescence. Then we also analyzed this gene expression signature. We also looked at it into human tissues. We found a similar pattern to what we observed with the genes based on genetic manipulations. In other words, you find an overrepresentation of genes overexpressed with age in human tissues, but not all human tissues, and with some exceptions. Again, the uterus is a very strong exception. So there are some exceptions, but by and large, you see again this increase in expression with age in genes associated with cell senescence in human tissues, in most, but not all of that. And then we did some network analysis. So we use co-expression data. I mean, as a side point, one of the things we developed some years back is this tool called GeneFriends, which is essentially a human co-expression map. So when we look at co-expression, well, we look at transcripts. So we have over 80,000 transcripts in the human genome, and you can look at how strongly they are co-expressed with each other. So basically, we take all of the gene expression data we can get our hands on, which is thousands of data sets, and then we combine it and we look at how strongly to genes or to transcripts are co-expressed. So basically, we have a matrix of 80,000 by 80,000, and how strongly each of them are co-expressed. Because genes that tend to function in the same process and pathway, they tend to be co-expressed together. So then we can use this for various analyses. So for example, if we have, we can build networks with it, which is what I'll show in the next slide. If you have a list of seed genes associated with any process or disease, you're going to look at other genes that tend to be co-expressed with them and identify new candidates. If you have a gene that you don't know the function of, you can look into which genes it is co-expressed with, and that may give you insights or ideas for potential processes that gene may play a role in. So it allows you to generate hypotheses using these data-driven methods. So, I mean, we published this and invite you to visit the database if you're interested. But basically, we use this co-expression data from GeneFriends to create a cell senescence co-expression network, which is what you see here. So we take the genes from cell age, the 279 genes, then we look at their interaction partners, so what they're co-expressed with, and then we build a network, which is what is here. And as you can see, there's different clusters, which we then color in different colors. And each color, I don't remember exactly which color reflects what, but they're basically processes that you'd expect. Cell division, cell cycle regulation, DNA repair, immune system. So each of these clusters would be a different function, a different process. So these are, in a way, this is a way of visualizing different processes associated with cell senescence. Then we did various analyses. I don't have time to go into detail, but we also combined the gene expression data. We used various network metrics, like we're trying to look for hubs, genes central in a network, trying to look for genes that connect different parts of the network. So I did various statistics on it. The point was that we did various analyses of network parameters to identify new candidates associated with cell senescence. So we settled on 26 candidates based on slightly different criteria actually. And then we tested this experimentally in collaboration with Cleo Bishop at Queen Mary's University of London. So this is the ones you see here. So these are the 26 candidates. Well, on top is a negative and positive control. Then you have the 26 candidates. And then what they've done is they silenced the genes and then in cells, and then they looked at various parameters, readouts associated with cell senescence. Cell number, cell area, markers of of cell senescence like P16 and P21. And as you can see, most genes, actually, when you silence them, they do appear to induce some biomarkers of cell senescence. So of the 26 genes tested, 6 of them are strong hits. That is, they induce significant and multiple biomarkers of cell senescence, and 12 of them are moderate hits. That is, they still induce some biomarkers of senescence. So that's most of them. I thought that was quite neat. I think that's a quite good example of this network biology approaches to derive candidates, which you then test experimentally, and they do seem to have some role in cell senescence. So, I mean, we published this last year. As I said, I mean, Robbie was a co-first author, and so this was an analysis involving many different people and also experiments in the lab. So there's various different types of results. I thought it was quite comprehensive systems biology analysis of cell senescence. So with that, I mean, so what are the main conclusions of our cell age database and analysis? So it's the first comprehensive database of cellular senescence. And as I said, we're working on a new version, which should release soon, that has more than twice the number of genes at the moment in cell age. Then genes promoting senescence tend to be upregulated with age in some tissues, not all tissues, but in more tissues than you'd expect by chance. Likewise, gene expression signatures of cell senescence are also upregulated with age. Again, in more tissues than expected by chance, but not all tissues, and there are some exceptions like the uterus. And then we did various network approaches of cell senescence and various analyses that we did more, so there's more analysis in the paper than I can mention here. And this reveals processes and new genes associated with cell senescence. So with that, let me just, so what's the road ahead for cell senescence? So I see cell senescence, I should say, as a mediator, first of all, as a mediator in aging. I don't see it as a primary cause of aging, but more as a mediator in aging. The other thing that's been, now emerging is this idea that senescent cells are actually important. They have important functions. There's been some really great work by Dimitri in Nice. Basically they show that if you ablate senescent cells, the mice die younger. So you kill mice if you do that, certain types of senescent cells. So it seems that senescent cells play some role. I mean, it's thought that senescent cells are an anti-cancer mechanism, but they also play other roles, including in development. And there's some nice data from Monroe Serrano on that. So I think that it's quite interesting that on one hand, they seem to have different functions. On one hand, they are associated with aging, but on the other, they also play important functions. And my interpretation is that they're mediators. They signal damage. They signal that something is wrong, but they're not necessarily the cause of what is wrong. That's kind of the hypothesis I've been going with. But I think it's something that it's quite interesting, really, what is the role. I don't see senescent cells like some others in the field, see them as like the root of all evils. Everything that aging emerges from them. I don't see them that. I mean, C. elegans and Drosophila, they age and they don't have senescent cells. So, we have to, have some balance on how we see senescent cells. But understanding their normal functions and how they're working or how their function or dysfunction contributes to aging, I think that's really the challenge. And so I think that's it. Running on time. Oh yeah, and I managed to dig up a couple of old photos of Robbie when he was in the lab, when we were both young. Well, thank you for the time and attention.
Thank you, Pedro. Nice pictures of me. I forgot about them. But yeah, we had great times there. I liked your hypothesis at the end. So would you think that a form of inducible treatment or modulated treatment, when you need to reduce the population of senescent cells, would be something more productive than the current approaches, which are to basically target all the senescent cells?
So yes, I mean, I see senescent cells a little bit like inflammation. I mean, if you think about inflammation, I mean, you cannot live without inflammation. It's very important, right? You need it for immune responses. You need it for repair. You need it for development. You need it for fighting infections. But too much inflammation in some circumstances is bad for you, right? So that's how I see senescent cells as well. Yeah, if there's situations where it becomes too much, there's got to be a Goldilocks zone. If it's too much, it becomes detrimental, just like inflammation. Now the question is, but not the question. The problem we have is that the tools that are at disposal are still too blunt. I mean, to control senescence. I mean, even to control inflammation. So I think that's part of the problem. You're right. If you can be more specific, and if you can control senescent cells or inflammation in very specific tissues. So, for example, inflammation is thought to contribute to some cancers. It's thought to contribute to Alzheimer's. I mean, so if you can control it in those organs, in those specific tissues, then yes, that would give you an advantage.
Okay, cool. I was going to ask regarding the list of genes that you presented.
There's also been, sorry?
I think we have some issues with the connection. Yeah, no, now you hear me well, right? Yeah.
So what they were trying to do is trying to reprogram senescent, developing approaches to reprogram senescent cells. So instead of destroying them, which can be detrimental, program them, make them healthy cells again, which I think would be more beneficial. But that's easier said than done. I think in practice that's quite difficult.
Mm-hmm. And I wanted to ask you regarding the list of genes that you tested experimentally in our paper. Did you do anything to follow up? I don't know, maybe in the direction of senolytics for any of those genes?
Not really, no. I mean,
No.
I have to be honest. I was happy we got so many hits, and yeah, cool. We're gonna have a nice paper. We never really followed up on it. I think when you look at the genes that were in the candidates, I mean, a lot of them are cancer related. I mean, they're important in cell cycle. So I think a lot of them are cancer related. Yeah, actually, I should go back and look at it in more detail.
Okay, thank you. Good. Any